Okay, welcome back. We have finished in the last module. We have seen various methods for pairwise sequence alignment. In particular, the focus of the last module was to look at exact methods for giving optimal pairwise sequence alignment. In this module 4, we are going to look at heuristics for pairwise sequence alignment. So, this lecture will focus on heuristics for genome scale alignments. So, we have finished 3 modules and today we will be starting the 4th module of this course, that is heuristic methods for pairwise sequence alignment. And in this lecture what we are going to do is we are going to look at first the motivation for why we need heuristics. Second, I am going to give you an overview of how these heuristics typically work, and in specifically we are going to look at 2 techniques. One is sampling minimizers, and in the next lecture we will look at maximal unique matches. So, these are 2 common heuristics that are used in widely used bioinformatics tools. So, let us start with the motivation for why we need heuristics for aligning sequences. Now, remember that for the various methods that we have seen in the last module, those are meant for pairwise sequence alignment. So, if you have 2 sequences, one of length m and the second sequence is of length n, then all of these dynamic programming based methods that we have discussed so far, they all require quadratic runtime, that is they have order m n time complexity. So, this time complexity can sometimes be computationally prohibitive when we work with real world data. So, I am going to give you one example. So, remember that when we are doing whole genome sequencing, we have a target genome that we want to sequence. Now, when we run this DNA sample through the whole genome sequencing pipeline, as output of the workflow we will get short fragments of the genome. So, we will get large number of these short fragments, and each fragment can vary from length 100 to few thousand bases. So, now we will get many these fragments of the genome, and now to understand what the unknown genome sequence was and also to know what mutations that unknown genome sequence has, we would typically align these reads, that is these fragments which I am calling as reads. So, we would align these reads to a reference genome, and those alignments would convey what mutations exist in the target genome. So, for example, if I see many reads aligning to this region, and suppose all those reads are saying that there is a instead of a T here, then I know that in my target genome that I want to sequence, there is a character instead of a T character in my unknown genome. Similarly, there is another mutation T instead of G at this location. So, these alignments give us the information about what mutations exist in my target genome. Now, when we work with real world data, we have to understand the amount of data that we are dealing with, right. So, for example, in a real world application where these alignments may be useful, for example, this is a real scenario where I am showing you visualization of the alignments to the reference human genome, and in this case these reads are derived from a patient that have, so the patient has cerebellar hypoplasia and spinal muscular atrophy disease. Now, the mutation that explains this disease is a homozygous mutation in TRP4 gene of the patient, and that mutation is reflected through the alignments of those reads to the reference genome. So, normally in a diagnostic workflow we would sequence the patient, we would align the reads to the reference, and those alignments would tell us what causal mutations explain the disease that are present in the patient. So, this is one real world scenario, and this is a common practice in many clinical services where they would do this process routinely. And now therefore, this alignment process has to be fast, because if they are generating billions of bases from the human genome, then the algorithms that they should have access to for doing this analysis should be fast and scalable. Let us look at a scenario where we will see that sometimes this order mn dynamic programming algorithms may not be suitable for dealing with large amount of data, and therefore, we may need heuristics at that point to make the process faster. So, to give a specific example, let us consider the example of Genome India project, and remember that before we discuss that, we have to remember that in the case of approximate pattern matching, we are solving the problem that is there a substring T dash of T. You can imagine that T can be the reference genome. So, I am looking for a region inside the reference genome that has small edit distance with my given read, that I am calling as pattern P. So, only if my read has a good edit distance to a region in the target genome, then we know that the read alignment exists. And we have already looked at dynamic programming algorithms to solve this kind of problem, and from there we know that these algorithms require quadratic time, that is in terms of the text size and the pattern size. So, this we have discussed in the last module of this course already. Now, let us consider the application of this algorithm in a real world setting where we have sequenced 10,000 Indian genomes. So, this is the Genome India project, which has recently completed sequencing of 10,000 Indian genomes using whole genome sequencing methods. And let us understand the amount of data that was involved in this project and see whether the dynamic programming method that we have discussed so far can be used at this scale. So, to do that, let us look at the statistics. So, as I said, Genome India project involves 10,000 human genome samples, and each sample will roughly have about 1 billion reads sequenced. Each read will have length 150 bases, and remember that each of these reads has to be aligned to the reference genome. So, in this case, the length of the reference genome is 3 billion characters, because we are considering the human genome reference. Now, we know that from our previous dynamic programming algorithms that these algorithms will require quadratic runtime. So, the complexity of the algorithm for each read will depend on the read length multiplied by the reference genome length, and that is for each read. So, for these many reads and these many samples, accordingly we will have to multiply. Now, let us consider how much time this whole process would take even if I use entire supercomputer available at IISc. So, in IISc we have Param Pravega supercomputer, which has 20,544 regular CPU cores. So, if I add up the CPU cores of all the compute nodes, then we have about 20,000 CPU cores running at frequency 2.9 gigahertz. And remember that when we are solving these dynamic programming matrices, so we have to fill the individual elements of these matrices, right. And remember that in the dynamic programming recursion, each value of the dynamic programming matrix depends on the neighboring elements of the dynamic programming matrix. So, each value in the dynamic programming table, we have to look for the value above, the value on the left, and value on the left upper cell. So, even if we use some instruction level parallelism, and let us be optimistic, let us say say that we can do 8 dynamic programming matrix cell updates per core per clock cycle. That is, in our dynamic programming table we can, let us say, solve 8 cells per clock per core, because in each clock we are doing these arithmetic operations, and in an optimistic scenario we can say that we are filling 8 cells of the matrix per core per clock cycle. So, this is the DP matrix that we had considered for the dynamic programming algorithms. Now, given this, I would like to analyze how much time it would take to do the entire computation for 10,000 human genome samples. Given that we have this much resources, that is, we have the supercomputing resources, and in these resources we can solve the dynamic programming table filling at this rate. So, let us do some maths to understand how much time it would take to solve the entire problem using the complete supercomputing resource. Right. So, let us first of all look at how many cell updates do we have to do for all 10,000 human genomes and for all billion reads of each human genome. So, for that we have to look at basically number of genomes times number of reads in each genome, because those many dynamic programming tables will have to. So, these are the number of times we will have to solve the dynamic programming table. Now, remember that if we look at standard practice in which we would use a fine gap penalty function, there remember that we were filling 3 tables that we had denoted using G, E and F. So, if we fill 3 tables G, E and F, then we have to multiply 3 accordingly, because we are filling 3 tables for each recursion, and the table size is basically the read length, that is 150 times the length of reference genome. So, if we put all the values here, what you can do is, so 10 raised to power 4, because that is the number of genomes we have, then 10 raised to power 9, because each genome will have about billion reads, times 3, because we are filling 3 tables, times read length, that is 150, and the length of reference genome, that is 3 billion bases. Now, if we add up all these values, what we get is the value shown on the left, that is 1.35 multiplied by 10 raised to power 25 cells updates. So, if you accumulate the number of cells in the dynamic programming tables that we are computing over the entire data, then these are the number of cell updates that we have to do to solve the alignment problem. Now, on supercomputing machine, if you look at the number of cores we have and the number of cell updates we can do per clock cycle per core, then as I said, roughly we had 20,000 cores. Each of these core is running at 2.9 gigahertz frequency, which means that it can do 2.9 billion cycles per second. So, these are the number of cycles at which these cores are running, and then we can also do 8 cell updates per clock cycle on each core. So, we can also multiply this by 8. So, this is 9, and if we do the maths, then what we get is this number. So, now if I want to analyze the total time it would require to complete the dynamic programming process, then I just have to take the ratio of these two numbers. So, this will be my numerator and this will be my denominator. because these are the number of cell updates I have to do, and these many cell updates I can do per second on the entire supercomputing machine. So, if we do the division, then we will get the number of seconds it would take to do the entire dynamic programming process. And what you get by taking the ratio is 10 raised to the power 10 seconds approximately, which is equivalent to 1000 years. And certainly we do not want to wait for 1000 years to finish this project, and that is why we need faster heuristics to deal with real world data. So, these heuristics will be still require lot of time, but that in practice will be maybe a few hours for each genome. So, on a single compute node. So, therefore, the topic of this module is to look at what faster heuristics we can use in practice to deal with real world data. So, let us get started. Now, intuitively, how will these heuristics work? Well, if you think about it, we have looked at two ways of doing pattern matching. One is approximate and one is exact. What we have found so far is that in the approximate matching, all these methods required quadratic runtime. Whereas, when we were discussing the exact pattern matching problems, we were able to solve these problems much faster. So, even without any index, we can solve this problem in order p plus t using Z algorithm that we have discussed so far. And if we consider indexes like suffix arrays, suffix trees, then we can achieve much faster times than this. So, the runtime that we have, for example, using suffix trees is only order p for solving a problem like this. And you can think that this is a significant gap between the two, that is, we can solve these exact matching problems significantly faster than approximate matching problems. And we have to use this idea in designing heuristics, that is, we can use exact matching first maybe and use those exact matches to guide how should we accelerate approximate matching process. So, let me give you a concrete example. So, in the approximate matching problem, we are solving this dynamic programming like this, where we have a table of size p cross t and we are computing these entire tables. Now, as some of you may guess, one way in which we can accelerate the dynamic programming process is maybe by looking for exact matches between pattern and text. So, if you can look for those short exact matches, maybe they can give us hints on where the pattern may align in the text. So, maybe as a first step, let us consider where the short matches exist between the pattern and the text. So, for example, in this illustration on the top, I am showing you exact matches of length 5. So, this is illustrating an exact match of length 5 between the pattern and the text. Suppose we look at all such short exact matches of length 5, then maybe these exact matches can guide us where a good alignment of pattern may be in the entire text. So, as the next step, what we can do is maybe we can confine our attention to those regions in the text T where we see a good cluster of matches. So, for example, as an illustration, maybe this is the region in the text that we want to focus, because we see a good cluster of matches here, and maybe if we focus on this region, we will be able to find a good alignment specifically in this region of the text. So, this is a rough idea of how these heuristics are going to operate. That is, we are going to use exact matches and then we will confine the attention in those regions of the text where sufficient number of exact matches occur. right. Okay. So, let us formalize these ideas next. Now, what do I mean when I say good cluster of matches? Well, we will mathematically formulate the clustering problem in a later lecture, but let me illustrate again using a figure. So, suppose we have identified the exact matches between the pattern P and text T. So, this time I am putting my pattern on x axis and I am putting my text on y axis. Now, these I am denoting these exact matches with these blue line segments. So, for example, this exact match conveys that there is an exact match between pattern and text, and you can imagine that there will be many exact matches that will occur, some because of there is a good alignment and some will occur by chance. So, once we have these exact matches, we have to narrow down where should we focus in the text, that is what is a promising region in the text where we should look for the alignment. So, as the next step what we can do is we can look for a good cluster of those matches, and roughly as I said, those exact matches should roughly form a diagonal like structure, that is we should be able to combine those exact matches to get a subsequent alignment. So, in that sense they should occur in a linear increasing order. And once we have found a good cluster of those exact matches, we will basically solve the dynamic programming table in only those restricted regions. Basically the goal would be to combine these exact matches by filling the gaps between two adjacent exact matches. So, using those exact matches as guiding regions, we can fill the dynamic programming cells around those exact matches and get the final alignment. So, our basically the dynamic programming computation is restricted to those regions where we have found a good cluster of exact matches. Now, we will formalize this clustering problem in a later lecture. For now, I just want to give you intuition of how this clustering will roughly look like and why this will help us accelerate the dynamic programming process. So, this way we are skipping the entire table computation and we are only focusing on those regions where those exact matches are located. Okay. So far I have given you the motivation for why we need heuristics, and I have given you a high level of overview of how these heuristics are going to work. Now, as a first step, let us look at some of the heuristic techniques for finding exact matches, that is basically this step. How are we going to look at these exact matches between the text and the pattern P? And basically we have to be clever at this step also, because we need a fast method to compute exact matches between the text and the pattern. Only then we will be able to move to the next steps. Okay. So, the technique that I am going to study now is known as minimizer technique. So, this is sampling minimizers, and later in the next lecture we will look at another technique which is known as maximal unique matches. Now, there are many techniques in the literature besides this as well. So, these are not the only two techniques that are present, but the reason I chose these two is because they are widely used in popular methods. So, for example, if you use some of the highly cited alignment tools, then these two techniques are one of the commonly used techniques for finding exact matches between the two sequences. So, for this lecture we are going to focus on minimizer technique. So, let us get started with what do we mean by minimizers. So, let us see. Now the simplest thing that we can think of to compute exact matches between two sequences is to consider fixed length words between text and pattern. Suppose that fixed length is 3. So basically we can look at all k-mers of length 3. So I can take my text, I can extract all 3 length words. In this case it would be CGT followed by GTT followed by TTC and so on. So I can pull out all 3 length words from text T, and similarly I can pull out all 3 length words from the pattern P. And once I have considered all the k-mers that are present in these text and the pattern, I can then subsequently see where are the matches happening. So for example, there is a k-mer match happening here. Remember that GTT can occur somewhere down the sequence as well. So then this will also be a k-mer match, right. Similarly you have a k-mer match between TTC and so on. So we can basically extract k length words from text and pattern, and then we can look at all the matches between text and pattern in the form of k length words. So eventually this will give us matches between the pattern and text. So all k length words in the pattern that have a match in the text will give us the matches between these two sequences. Now it turns out that for desirable values of k, for example I can choose k equal to 30 or k equal to 50 in practice, because of repeats in the genomes it often leads to too many matches in practice. So the number of matches that we find in practice can be too many to process. So if I consider human genomes for example, so if you think of human genome versus chimpanzee genome, and if you pick k equal to 31, then you are going to find billions of k-mer matches between those two genomes in practice. And therefore we have to be slightly clever in how we are considering these k-mer matches. So in order to effectively process the downstream stages, we have to find a way to reduce the number of matches effectively. Now one common technique is to sample the k-mers. So can we sample the k-mers, and one popular technique to do that is known as minimizer technique. Now the way this minimizer technique works is following. What we are going to do is, again we are going to consider all k length words, that is k-mers from the sequence, and I am going to use a random hash function that will basically map my k-mers to integer. So between all k-mers, k length words, I will map it to a integer. And let's consider let's use any random hash function. So what I am going to do is I am going to hash each k-mer, and I am going to get an integer representation of that k-mer. Now with that, so once we have these integer values, the way minimizer technique works is by considering a fixed length window of size w. So we will look at consecutive k-mers in that fixed length window. So in this example I am going to look at 4 consecutive k-mers, and I am going to select the hash value which has minimum value. So I am going to pick k-mer which has minimum hash value. In this example you can see that 4 is the minimum hash value. So therefore I am going to select 4. Then I am going to slide this window one at a time. So I am going to slide this window further, and I am again going to check what is the minimum hash value. So this time the minimum hash value is 2, so I am going to select 2. Then I further slide my window, and again I am going to check my minimum hash value. this time the minimum hash value stays same. So, I am not going to pick a new k-mer. Further, I slide my window, this time the minimum value has changed and I pick a new k-mer which has hash value 1, and subsequently I continue this process until I reach the end of the sequence. So, basically effectively you are picking minimum values from each sliding window, and therefore, a k-mer would be sampled if it is if its hash value is minimum in at least one of the windows overlapping with it. So, for example, in this example we have selected 3 k-mers. So, out of all the k-mers in the sequence in this particular example, we have sampled 3 k-mers, that is this k-mer right here, then this k-mer and then this k-mer. So, instead of selecting all k-mers, this technique allows us to sample k-mers using this sliding window minimum idea. So, the parameters to this algorithm are 2, that is the value k, that is what k-mer length you are considering, and the value w, that is what window length you are considering, right. So, based on these 2 parameters you will get a final output in terms of the sampled k-mers from the sequence. So, okay. So, let us proceed forward. So, why is this technique helpful? First of all, okay. So, before that we have to see that this is trivial to process, because all we are doing is sliding this window one by one and picking the minimum value in each window. So, if I am considering sequence S, then basically I will run a for loop, that is running a for loop over the entire sequence, S, because I am sliding this window one by one over entire sequence, and then maybe I need a second window, that is a second for loop to just pick minimum in that window. So, I need first for loop to process the entire sequence and the second for loop to process the minimum value in a particular window. So, therefore, because the window size is w, this is doable in order s times w time, and it is a trivial algorithm, that is it is a simple code which requires 2 for loops. So, now let us understand why this technique is useful. So, the reason why this technique is useful, because now what we can do is we can sample k-mers from both text and pattern, and once we have sampled k-mers from both text and pattern, we can then look at the matches between the minimizers only. So, these matches will correspond to the minimizers that are common in text and pattern both. So, now because we are considering a subset of k-mers from text and pattern both, we will have fewer matches to process, right. So, this allows us to reduce the runtime of the subsequent steps and improve the scalability of this alignment process. Let us also analyze the properties of why this what is the advantage of this minimizer technique. So, the first useful property of this minimizer technique is the window guarantee, that is, if 2 sequences share a common substring of sufficient length, then they are guaranteed to sample a common minimizer, and this property is easy to see, because if they have a common substring of length w plus k minus 1, then basically what we are saying is that these 2 sequences have a common window where there are equal w k-mers. So, the w k-mers that are present in these 2 windows are equal, and because these k-mers are equal, the minimum value that I am going to select on the sliding window that is present here and the sliding window that is present here is going to be equal, because the k-mers that are present here are equal. Therefore, the minimum value that will be selected is also equal. Therefore, it is guaranteed that we will have a common minimizer selected in this region. So, if the minimum value selected here is this, then the same k-mer will be selected as minimum here, and that guarantees us that we will consider the exact match between these two regions. So, we will not miss a match between these two common substrings of T and P, and that is important, because if there is significant similarity here, then we want to make sure that we have matches, because ultimately we are going to use these matches to do the alignment, right. So, remember that these matches will be basically used to do the alignment as well. So, this window guarantee is useful to ensure that we have matches present in the common substrings of the two sequences. Okay. So, this property is easy and it is useful. The second important property of minimizers is following: the expected fraction of minimizers selected from all the k-mers given a random input sequence equals 2 by w plus 1. Basically, what we are saying here is that the longer your window size is, so remember that w is equal to the window size. So, the longer your window length is, the fewer is the minimizers that you will pick. So, the expected fraction of minimizers sampled is inversely proportional to the window length. Now, why is that the case? Well, that we will have to prove mathematically why is this case, why is this true. But from a practical perspective, what you can see here is that we can control the sampling rate by selecting appropriate value of w. So, for example, the commonly used values of w are 20 to 100, and with these values you easily see that we are getting a very good sampling ratio. So, with a value of 100, that means that on an expectation we are going to sample 1 by 50 of all the k-mers that are present, right. So, let us mathematically prove why is this the case, why is the sampling rate or why is the expected fraction of minimizer selected among all the k-mers is 2 by w plus 1. Okay. So, let us try to prove this claim, and the claim is following. Now, let us start the proof. Let wi be the window of w consecutive k-mers starting at position I. So, if my sequence, if I am considering a sequence S and I have a position I, then I am considering the k-mers that are present in the sequence, and I am calling wi as the w consecutive minimizers present in this window, right. So, this is capital W. So, wi denotes the w consecutive k-mers starting at position I of the sequence S. Next, let me also denote ki as the k-mer starting at position I in the sequence S. Now, let me also define something known as charged. So, the window wi is said to be charged if wi selects a new minimum value, right, because remember that when we are sampling minimizers, we will say that Wi is charged if Wi selects a new minimum value. If it uses the minimum value from previous window, then we will say that Wi is not charged, but if it selects a new minimum, then Wi is charged. So, what we have here is basically the expected fraction now can be analyzed by counting the number of charged windows in the input sequence, right. So, all we have to do is to look at the number of charged windows, because only those windows we have to consider where we are picking a new minimum value. As we are sliding window one by one on the right, we have to look at those windows where a new minimum value has been selected, because that will give us the fraction of k-mers that have been selected as minimizers. So, in the next step we will focus on counting the number of charged windows in the input sequence. So, continuing the proof, let me also define a random variable xi, which is 1 if window Wi is charged and xi is 0 otherwise. Let us compute this value E of, let us compute the expected value of xi. So, to do that, what I am going to do is I am going to look at w plus 1 consecutive k-mers. So, if I consider w, this my window Wi, where I had w consecutive k-mers. Remember that. So, the definition of Wi is that we have in the Wi window we have w consecutive k-mers, right. So, now I am going to consider w plus 1 k-mers, which is basically I am also going to consider the k-mer just before the window. So, these are the w k-mers and this is the additional k-mer that I am going to consider now. So, this will be, if this is position I, then this is ki minus 1, this is ki and all the way to ki plus w minus 1. So, these are the k-mers I am considering. Okay. Also, let us say that k min is the k-mer with minimum hash value among these w plus 1 k-mers. So, this is the minimum. So, this is the k-mer with the minimum hash value among these w plus 1 k-mers. Now, observe that when will this window Wi will be charged, that is when will this window Wi pick a new minimum hash value. It will pick a new minimum hash value in only two scenarios, that is when k min equals to ki minus 1, that is so that is if my minimum value is among these w plus 1 k-mers is this value, or if my minimum value is the rightmost value. So, only in these two situations I am going to pick a new minimum value in Wi. So, in window Wi will be charged only if k min equal to ki minus 1 or k min equal to the last k-mer, that is ki plus w minus 1. If any other k-mer is minimum value, then basically remember that if any other k-mer is minimum, then it will be also be minimum value of the previous window, because if you look at the previous window, then that particular k-mer will also be the minimum of the previous window. So, in that case the new window Wi will not be charged. So, basically when we are computing the expected value of xi, we have to only consider two scenarios, that is k min equal to ki minus 1 or k min equal to ki plus w minus 1. So, to reiterate, the window Wi will be charged only when the last k-mer is the minimum value or the k-mer just before the window is the minimum value among these W plus 1 k-mers. So, only in these two situations is when window Wi will be charged, that is window Wi will pick a new minimum value, because if this is the minimum, then as we slide we will have to pick a new minimum value, and if this value is the minimum value, then as soon as my window is shifted, this will be the new minimum value of the new window. So, I encourage you to think about it, that these are the only two cases in which Wi window will be charged. And if you agree with me, then the expected value of xi will be nothing but the probability of k min being equal to ki minus 1 and the probability of k min being equal to ki plus w minus 1. So, because we are using a random hash value and there are w plus 1 k-mers, so this will happen with 1 by w plus 1 value, and this will also happen with 1 by w plus 1 value, because we have w plus 1 k-mers. So, any k-mer being minimum hash value will be equal to 1 by w plus 1 probability. So, adding this two terms we get the factor 2 by w plus 1. Now, in this proof we ignored the possibility of ties, that we assume that all these w plus 1 k-mers are unique, and that is a safe assumption because we are considering a random input sequence. If you look at the space of all possible k-mers, that is sigma raised to power k, that is significantly higher than w. So, the probability of a tie is extremely small, because my window length is much smaller than the space of all possible k-mers. As a result, we can ignore the possibility of ties and we can assume that all these w plus 1 k-mers are distinct, and therefore, we get these probability values 2 by w plus 1. So, this completes the proof, and here we have managed to prove that the expected fraction of minimizers that will be selected from all the k-mers in a random input sequence is 2 by w plus 1. That is, in other words, if your entire sequence has, let us say, alpha number of k-mers, then this minimizer technique, the number of sampled k-mers will be 2 alpha divided by w plus 1. So, the fraction basically that you achieve is 2 by w plus 1 using minimizer sampling, and this is why, because now we have managed to sample the k-mers, we basically are able to reduce the number of matches when we are comparing two sequences. So, this is one reason why minimizer technique is useful, because it helps us speed up the alignment process as we have fewer matches to deal with. So far we have looked at how this sampling technique works. We have looked at a popular technique known as minimizer technique for sampling k-mers and how that helps to compute exact matches between two sequences. In the next lecture, we will also look at one more popular technique for computing exact matches, which is known as maximal unique matches, or MUMs in short. And this is, once we finish the topic and once we know how to compute exact matches efficiently, we will then move to clustering formulations, where we will see methods to cluster these exact matches and get a good alignment in the end. Thank you.